Killing cancer cells is easy. The difficult part, not harming normal cells in the process. To that end, we must understand the biological differences between cancer and normal cells. One such fundamental difference is their chromosome composition. All normal cells in our body contain 2 sets of 23 chromosomes, one from each biological parent, 46 total. In contrast, cancer cells contain abnormal numbers, either more or fewer than 46. This phenomenon is called aneuploidy, and it has been known as a hallmark of human cancer for over a century. And yet, we know very little about how aneuploidy promotes cancer development and progression, and we still lack drugs leveraging this fundamental difference to treat cancer. My lab applies experimental and computational approaches to address these challenges. We strive to understand the causes and consequences of aneuploidy and devise strategies to selectively kill cancer cells by targeting aneuploidy. In a recent study, we mapped aneuploidy landscapes of about 1,000 human cells. Then, we compared the weaknesses of the most and least aneuploid cells to identify where they might be the most vulnerable to drugs or genetic therapy. Among other findings, we identified a specific protein, KIF18A, that we find to be dispensable in normal cells, but essential in aneuploid cells. Then, we showed why, biologically, aneuploid cells are more sensitive to our inhibiting of KIF18A, and, in this way, weakening and killing the cancer. We published this work in the journal Nature in 2021. In parallel to our work, 2 pharmaceutical companies developed KIF18A inhibiting drugs, but didn't know which patients these drugs should be tried on. Following our work, and that of colleagues who reached similar conclusions, they decided to test their drugs on patients with the most aneuploid tumor types. It's exciting, our work has already affected clinical trial design, and we hope our findings pan out in these patients. This is just the tip of the iceberg, though. The more we understand aneuploidy, the more we can harness this basic cancer trait to benefit patients. The future of cancer treatment lies in precision cancer medicine, and aneuploidy is poised to play a major role in this future.